A big hello to all my wonderful and enthusiastic NEET aspirants out there. I hope no one has lost their enthusiasm in the whole process of NEET preparation, my dear children. Please make sure that you are happy, you are healthy, then only you can study well. Okay, let's have that positive attitude and let's ace NEET. And also remember that this particular channel is like a small baby which has just been born. And what do you do to the small baby? You motivate them. How do you motivate them? You say that, yeah, yes, you can walk, you can do this, come here, go there, things like that, right? Similarly, we also need lots and lots of motivation, my dear children. For that, I'll recommend you, please definitely share the videos, like the videos, comment down whatever you think. Okay, am I teaching bad? Am I teaching good? Or if you have any doubts, put it down. We are here to solve. I think all the comments have been replied to. Okay, it's not like we did not. And it's not like we just have this particular channel to look after. No, there is another big channel out there, Deeksha Karnataka. And we are the small sister channel that we have formed over here for NEET aspirants. So we want your support. Okay, so make sure you are giving enough amount of support to the small child which has been just born. Yes. Now going ahead and hoping you would be understanding things, let's start with first question. Match list 1 with list 2. So over here Rhizopus, Cystilago, Puccinia as well as Agaricus is mentioned, and over there you have to match it with this. So I know one very important thing. Rhizopus I have been studied from class 10th itself, that or before that. Okay, Rhizopus is the one that causes bread mold. Okay, the bread turns green black in color because why? There is formation of Rhizopus. Okay, so this is known as bread mold. Rhizopus is known as bread mold, and A goes with 3. A goes with 3. I just have 2 options like that. So let's exclude these 2 options. Okay, can I exclude these 2 options? Okay, now going ahead. Agaricus, my dear children, I'm pretty sure it is known as mushroom. It is what? Mushroom, my dear children. So D goes with 1. D goes with 1. I just have 1 option, and with that I get the answer as well. Puccinia is something that causes rust. Okay, that is known as rust fungus as well. It causes diseases in plants. And Cystilago is known as smut fungus. Cystilago is known as smut fungus. Okay, Puccinia or Puccinia is known as rust fungus as well. Okay. Works. So we have to solve questions within 60 seconds. That is how you have to train yourself in examination. Now going ahead with the next question. Identify the part of the seed from the given figure which is destined to form root when the seed germinates. So when the seed germinates, what is that part that will be giving rise to the root, my dear children? And this is what? This is a monocot seed. How do you understand that, ma'am? Monocots are the one that has 1 cotyledon. Oh, does not have 2 cotyledons? No, that is there in dicots. And how do the dicot make up the cotyledons? They take the endosperm and they make up the cotyledons out of it. But over here, they have little endosperm still left back, which is why they just make 1 cotyledon. So this is the persistent endosperm which is there. That is why the picture is like this. And then they have their own cotyledon as well. Now you are asked, what is that structure that will be giving rise to root? Root always comes from something that is known as radicle. And what is radicle over here? This part that you see over here, this is radicle. My dear children, always remember radicle is covered by root cap. And in the case of monocots, they have an extra layer down there. Okay, down there, which is D. What is D? Coleorhiza. What do you call it? Coleorhiza. Okay, there is coleoptile at the top. Coleoptile. Probably at the top, yeah, and coleorhiza has an R thing, an R for radical, so probably it's down. Okay, remember like that. So in between the coleorhiza as well as the radical, what do you have? You have the root cap. Just for your understanding, I just told you. Fine. So for root you have radical, for shoot you have plumule. Over here you asked about root, so your answer becomes C. Cool. Yes. Now lecithin, a small molecular weight organic compound found in living tissues, is an example of. Lecithin, my dear children, it is a lipid. It is a lipid. Okay, ma'am, then this option goes off. Amino acid is basically protein. Amino acid makes up the protein. It's a whole different biomolecule. Carbohydrates, ma'am, it's again totally different. Phospholipids and glycerides. Basically, both of them are lipids. Glycerides basically means what? Glyceride basically means a combination of glycerol plus fatty acid. Triglyceride, diglyceride and things like that. Remember? So this is actually forming fat. That is a lipid, right? And this is phospholipid. What is the difference between that? There's a phosphate group added to it. Ma'am, you said lecithin is a lipid, and in which category does it come? The answer is phospholipid. It has a phosphate group, which is why I'll call this as phospholipid, my dear children. Lecithin is a small molecular weight organic compound found in living tissues, and it is an example of phospholipids. It is very important in the formation of cell membrane and things like that. Okay, so your answer becomes phospholipids. Will you forget it? Lecithin, phospholipids. Now, match list 1 and list 2. Or list 1 and list 2. Okay, fine. 2 or more alternative forms of a gene. two or more alternative forms of a gene. That is, I have a gene coding for height. One is capital T, another one is small t. This codes for more amount of protein, this codes for less amount of protein out there. So both of them are different forms of a gene. What do I call them? I'll call them alleles. I'll call them alleles, my dear children. So your A goes with 3. A goes with 3. What option do I have? I just have one option, and with that I get the answer as well. So can I eliminate all these options? Let's see if all of them are matching or not. Now, cross of F1 progeny with homozygous recessive parent. What are you going to do? You are crossing F1 progeny, okay, the first generation progeny. Suppose I got this as a F1 progeny, okay, and now I'm going to cross it back with homozygous recessive parent. That is like this, recessive, it has to be recessive. So I'm going to cross it with this. This is known as what? Back cross? No. This is known as test cross. When you're specifically mentioning that you are going and trying to cross with a homozygous recessive parent, you're very specific about this. This will be what? A test cross. And why do you need a test cross? For understanding the genotype of this. Suppose I have got tall plants. This is a tall plant. This is a tall plant. This is also a tall plant. How do I differentiate whether this is tall like this or is it tall like this? I am going to cross it with the dwarf, homozygous dwarf plant or recessive plant, my dear children, and the product that I'm going to get from two of these things would be different, and on that basis I would analyze, oh, this plant that I'm seeing is actually tall like this, it's a pure tall, I can use this for hybrid programs. So that is why we do this, that is the test cross. So B goes with 4, yes, correct. Cross of F1 progeny with any of the parents, not specific, you're just going back, crossing with any of the parents, this will be known as back cross, this will be known as back cross. Can I say all test cross is back cross? All test cross is back cross? Yes, because anyway I have to go back. But all back cross is test cross? No. All back cross is not test cross. Suppose I'm crossing it with this. Is it a test cross? No. You get it? So that is the thing. So remember that also. Number of chromosome sets in the plant is known as ploidy. It is known as ploidy, and with that we get sure about the answer, that is 3. Yes. A transcription unit in DNA is defined primarily by 3 regions in DNA, and these are with respect to upstream and downstream end. Okay, so basically what are the 3 regions if you talk about transcription unit? First will be a promoter region. So promoter region over here, this will be 3' and this will be 5' and this will be 5' and this will be 3'. Where is transcription going to happen? Where is the RNA polymerase going to make a new DNA polymerase? RNA over here, over in this particular area. RNA polymerase sorry for the disturbance, RNA polymerase makes up a new RNA at this direction. So it comes join over here and then starts off this, not exactly here in the promoter region. So it is in this direction that it goes. What do you call this strand as? You call this strand as template strand, which is used as a template for transcription to happen. And on the other hand, you call this strand as coding strand. Now, in a transcription unit, you have a promoter region, and this region that you see is known as terminator. Terminator. Here, the process of transcription is going to terminate. What do you call this particular gene which is going to be transcribed completely? It is known as structural gene. Okay, that's my hand doing wonders. Just give me a second, I'll be right back. Don't go anywhere. Okay. Yes. So this is known as, why God? Why me? Okay. Structural gene. Cool. If you are asked where is the promoter located, you say in reference to the coding strand, you'll say it is located towards the 5' end of the coding strand. Where is the terminator located? It is located to the 3' end of the coding strand. Okay, not give reference to template strand. Remember that as well. So over here, the RNA polymerase along with the initiation factor, that is sigma factor, will come and attach. Okay, do the transcription. Okay, the sigma factor will go off after initiating, and then at the terminator region, rho factor will come and attach and terminate the whole thing. Rho factor, it want to terminate, that is why it's crying, so that's why it needs rho factor. Remember all this important piece of information. So whenever I am solving a question, I am also giving you something associated with it, so please remember it, and here comes your answer. Promoter, structural gene and terminator. Okay, not repressor, operator protein, this does not make sense over here. Yeah, that is not a transcription unit. Now, auxin is used by gardeners to prepare weed-free lawns, okay, weed-free areas, yeah. But no damage is caused to the grass as auxin is what? So when I put auxin, I am basically I am preparing the lawn for a weed free area. There is no growth of weed over there. But my dear children, weed means unwanted plants, right? Now, but whatever I do, it won't be damaging the monocots. Why? Why is that so? Because it promotes apical dominance. Yes, it does, but it does not make any sense over here. Apical dominance means it allows the apex to growth. Okay, it allows the apex to grow. It does not allow the lateral growth of plants. It does not allow the lateral growth of plants, but allows the apical growth. Now that is there. Promotes abscission of mature leaves only. It actually inhibits, okay, it actually inhibits, prevents abscission of mature leaves. That is also false. Does not affect mature monocotyledonous plants. Is that the reason? Grass is what? A monocot. And probably auxin is not going to affect the mature monocot plant, which is why there is no damage. Yes, so this becomes your answer. Can help in cell division in grasses to produce growth, my dear children. This is again a wrong option. Okay, cell division and things, it helps in cell elongation. Cell division is majorly a function of your cytokinin and things like that. So that is a thing. Okay, cell elongation happens, that is also part of growth, but not exactly over here. Cell division is being mentioned. I would recommend not to go with this. Please go with the third one. Does not affect the mature monocot plants. As simple as that. Now, going ahead. These are regarded as the major causes of biodiversity loss. Okay, so it is known as the evil quartet. I'll tell you. The major causes of biodiversity loss is categorized under evil quartet. That means there are 4 things. Quartet means 4. 4 things that are very evil and that causes the biodiversity loss. First is habitat loss and fragmentation. That means basically, for example, I am living over here, you're taking away the habitat, where will I live? Okay, there is a loss of habitat. Yeah, or probably this particular studio is cut into 2 parts. Okay, my fragmentation is happening and in this part something else is being done, a new building is being constructed. Can I teach? No. So basically all of this causes biodiversity loss. Then after that you have overexploitation. That means, what do you mean by overexploitation? Basically, there were certain organisms which were really good source of something. I'm going to kill all of them because I want that something. You get it? Probably I need food, which is why I'm killing it. But I'm doing it in an over manner. I'm not worried about anything that the number will be decreasing or what will happen. I'm just killing it randomly. This was happening in the case of stellar sea cow, which is native of Russia. That got extinct now. Okay, that got extinct because of overexploitation. Same thing with passenger pigeons as well. So overexploitation is one big reason. Now after that you have invasive species. Invasive species. What do you mean by invasive species? For example, I have a nice area. Okay, now in your NCERT there is an example of Clarius garipinus, which is a catfish. Okay, it was introduced from outside, but what happened was the native catfishes which were there, they were not able to live when this guy was introduced, because this guy was competing for everything. So all the native catfish species, they got extinct. So it is invasive. It is coming and invading this particular area, competing for resources and probably killing it. That is also a reason of biodiversity loss. Now the fourth one, the last one in evil quartet is what? Basically co-extinction. Co-extinction, my dear children. For example, one organism is totally dependent on other organism, my dear children. So basically, if that organism, suppose something happens to me, the other organism that is depending on me will also not be working. That is the thing. So it's not me and you thing. Okay, it's just generally the plants. For example, a plant is there, a pollinator is there. Okay, suppose this pollinator is not there, it is dead, it is getting extinct. What will happen? I will also get extinct because I cannot allow no one else to do pollination. This is how I used to have my pollination cycle going on. You get it? That is known as co-extinction. So these are the 4 things. So these are regarded as major causes of biodiversity loss. It should include all these things, otherwise there is nothing. So overexploitation, co-extension, habitat loss and fragmentation are the reasons. Do you see mutation and migration as a reason? No. They are not the major causes. I am not saying they are not the causes, but I am saying they are not the major causes of this thing. So I will say choose the correct option. A, B and D. A, B and D only. This becomes your answer. Cool. Will you forget this? Habitat loss and fragmentation, overexploitation, invasive species, co-extinction. It's just remember like this only. This studio getting half and things like that. Overexploitation, basically if you're overexploiting me, it's saying ma'am teach this, ma'am teach that, what will happen? I'll die. Okay, so invasive species, as in some other teacher comes in who is very much better than me, then what will happen? I will be extinct. There is no use of Jasmine ma'am then, right? Yes, so like that. Co-extinction, basically children, if you are not there, I am not there. That's the thing. If you're not watching this video, I am are not going to be of any use. It's not like they will remove me. Nothing will happen like that. But yes, this is a fun way of remembering things. If you want, you can remember in that way as well. Now, going ahead to the next thing. Match list 1 and list 2. List 1, list 2. Okay. Clostridium botulism. Okay, so over here I see the products and over here I see the organisms which are producing it. So Clostridium botulism, the name suggests butyric acid. Clostridium botulism is the one that produces butyric acid. It is very easy for me to spot. So let's see, A goes with 3. Do we have an option like that? A goes with 3. I have 2 options like that. So let's exclude these 2 options. Now, Saccharomyces cerevisiae, which is yeast, my dear children. Yeast does fermentation. And what is the product of fermentation? It produces alcohol. What alcohol? Ethanol. It produces ethanol. So B goes with 1. B goes with 1. I have 2 options like that. Okay, fine. Let's go ahead. Streptococcus. Streptokinase sounds similar? Yes, my dear children. So what is happening? Streptococcus is a bacteria that produces streptokinase. What is streptokinase? It is a clot buster. Please remember it. What does it do? It bursts down the clot. Okay, so there won't be any clots up there. So that is the use of streptokinase. Okay, remember that. So D goes with 2. I have just one option and this becomes my answer. Let's talk about C over here. Trichoderma polysporum. Trichoderma polysporum is a fungus and it produces cyclosporin A. Ma'am, what is cyclosporin A? What does cyclosporin A do? It is an immunosuppressive agent. That means, for example, I have a graft done. I am getting a kidney from some other person. Okay, that person is helping me. But my body thinks it's foreign and tries to fight against it and probably damage the kidney that I have just got. Is that fair? No. So what do I need to do? I cannot tell my body to work like that way, but I can basically give my body immunosuppressant that will suppress the immune system, which is why that particular organ that I have transplanted could survive in my body. So immunosuppressive agent, it is what? An immunosuppressive agent, cyclosporin A. I was thinking of way to make you remember this. Cyclosporin, cyclo. So initially we had a cycle and let someone else also, no, let's not make it complicated. It's easy that way. Okay, now next, spindle fibers attached to kinetochores of chromosomes during what? During what phase? Spindle fibers, my dear children, I hope you know that centrioles are going to produce some spindle fibers, and these spindle fibers are going to attach what? The kinetochores which are over here. When do they attach? During metaphase. During metaphase, what is happening basically? In the case of prophase, basically spindle fibers and everything, centromeres are going to that. So centrioles are moving towards the opposite poles, and they start forming the spindle fibers, yeah. But then later on in metaphase, they actually come and attach and they align them in one particular line, equatorial plane, my dear children. So that happens in the case of metaphase, not prophase, not anaphase, not telophase. In prophase, yes, the beginning of all the things happen. They move there, they start forming spindles but not attach. Attaching happens in the case of metaphase, yeah, and they arrange them in equatorial plane, and then later they are separated in anaphase, and in telophase you have all the nuclear layers, nuclear membrane, everything reappearing. That all went off in prophase, it came back in telophase. So your answer becomes metaphase. Works? If it works for you, it works for me. Now, which of the following is an example of actinomorphic plants? What do you mean by actinomorphic plants? Actinomorphic plants, this is an actinomorphic plant. Now you tell me the answer. This thing that you see over here, it's too much contrast. It is an actinomorphic plant. Probably that means that I can cut it into equal halves from any plane, my dear. If I cut it like this, I get equal half. If I cut it like this, I get equal half. So whatever I do, I get equal halves. In your animal kingdom thing, you might have studied about radial symmetry, radially symmetrical organism which you can cut from any plane that passes through the center into equal halves. Multiple planes that passes through the center into equal halves. So over here, Datura, Cassia, Pisum, Sesbania, all of them, they are the examples given over here. You have to choose what is actinomorphic. What is this particular flower? This is Datura. And in the question, the picture is not given. I added the picture for you to understand. This is Datura, my dear children, and this is actinomorphic. They can be cut into equal halves through any plane that passes through the center. Now, Cassia, Pisum and Sesbania, basically all of them are zygomorphic. Are what? Zygomorphic. What do you mean by zygomorphic? Bilaterally symmetrical. That means it can be cut into equal halves only through one plane, none of the other planes. What are we? We are bilaterally symmetrical. We are also bilaterally symmetrical. Similarly, these plants, Cassia, Pisum, as well as Sesbania, my dear children, they are all zygomorphic. So you got to understand two things from one question. Datura is actinomorphic and this all things are zygomorphic. Works? actinomorphic A for actinomorphic, A from all the planes. You can remember like that. Cool? Yes. Now, the cofactor of the enzyme carboxypeptidase is, what is carboxypeptidase, my dear children? It's an enzyme that would be cleaving around the peptide bonds towards the carboxyl terminal in the case of a protein, peptide, right? Now, the cofactor, you need cofactor. Certain enzymes need cofactor for their working as well. So, what is the cofactor for carboxypeptidase? It is none other than zinc. It is none other than zinc, my dear children. Please remember, carboxypeptidase has a cofactor and that cofactor is zinc. Without zinc, it won't be working. Cool? Now, over here I have written about a lot of things. Carboxypeptidase are exopeptidases, enzyme that hydrolyze peptide bonds at the C-terminal, carboxy terminal of proteins or peptides, releasing individual amino acid. It releases all the amino acid, okay? Now, why not the other option? Niacin, that is this. Vitamin B3, okay? NAD, it is a part of what? NAD and NADP, which acts as a coenzyme in redox reaction, not as metal cofactor. So, it is not a metal cofactor, and basically this niacin is a part of NAD and NADP, which acts as what? Coenzymes, my dear children. They are acting as coenzymes. Now, over here, flavin. Again, flavin nucleotides help in electron transfer. Example, in the case of succinate dehydrogenase, that is an enzyme unit, yeah, and it is totally unrelated to all this thing. Heme is found in peroxidase and catalase. Heme is a prosthetic group in enzymes like peroxidase and catalase involved in redox reaction, not in carboxypeptidase activity. So, basically, you just have one answer left, and that is zinc. Zinc is the only thing that will be acting as a cofactor for carboxypeptidase. Zinc and carboxypeptidase. You have to remember it. Okay? Now, match list 1 with list 2, nucleolus, centriole, leucoplast and Golgi apparatus. Let's see. Okay, nucleolus, I'll tell you, nucleolus is the place basically where our RNA is made. You know, nucleus has a major role in the formation of ribosome. And what is ribosome made up of? Ribosome is made up of rRNA, ribosomal RNA and things like that. So, obviously, it has to be linked with something like that. Now, centriole, if we talk like that, centriole are the one that gives rise to spindle fibers, and they have a cartwheel structure. They have what? A cartwheel structure. Now, leucoplast, basically they are colorless plastids that you have. They are colorless plastids, and if they are not colorful, then what's the use of this? Probably they are going to store something. They are probably going to store something. So, leucoplasts are there for storage. Golgi apparatus, my dear children. So, what does RER and SER do? You know, smooth and rough endoplasmic reticulum. Rough endoplasmic reticulum gives you proteins. Smooth endoplasmic reticulum gives you lipids. Okay. So, proteins and lipids now came to Golgi apparatus. Golgi apparatus will be beautifying it. Will be beautifying it as in it will be adding certain groups and making it very, very active and making it useful. So, usually, let's say the carbohydrate molecules, okay, some part of it would be added up there, which is why we'll now call it as glycolipid or glycoprotein and things like that. So, all this formation happens in the case of Golgi apparatus, and then later after forming it, it also packs it and sends it off. Okay, so these are the function. And with this function that I have told you, can you try to solve the answer now? So, nucleolus, my dear children, nucleolus, rRNA. So, over here I just see one, okay, site for active ribosomal RNA synthesis, that is nucleolus. A goes with 3. A goes with 3. Do I have an option like that? Yes, ma'am. I have this and this. So, can I cancel this and this? Yes, I can cancel this and this. Now, centriole, it is organization like a cartwheel. B goes with 2. B goes with 2. Over here, I just have one option where B is going with 2. Okay, see, leucoplast, my dear children, goes with for storing nutrients. For storing nutrients, my dear children. You have under leucoplast, you have elioplast, amyloplast and things like that. Amyloplast stores carbohydrate, elioplast stores oil, that is fat and things like that and things. Okay. Now, yes, and Golgi apparatus, my dear children, is site for formation of glycolipid. You added the carbohydrate group and things like that, right? The lipid. So, this goes with that, and this is how we are going to get the answer. Cool? Yes. Now, the capacity to generate a whole plant from any cell of the plant is called as, suppose there is one cell that can give rise to the whole plant. What do you call this ability or the capacity to generate a whole new plant out of it? This is known as totipotency. This is known as totipotency. So, in the case of plant cells, you know, meristematic cells are there towards the tip and things like that. These cells, if you're taking a part of it and letting it grow, providing nutrient medium, it will grow to a whole new plant. it will grow to a whole new plant, my dear children. And this particular thing is being used in tissue culture method, And in tissue culture method, when you are producing so many plants, okay, from little X plants, my dear children, you call this as micropropagation. In tissue culture only, when you are producing, you use tissue culture method for micropropagating lots and lots of plants. Okay, differentiation is a whole new thing. For example, I have a cell. This cell becomes mature and says that I am going to act like a parenchyma cell. So this is differentiation. This process is known as differentiation. The somatic hybridization means what? Basically, you have two cells. Okay, and you're going to hybridize these two cells, the protoplasm of these two cells. Okay, if you're removing the cell wall of the plant, what is left behind is known as protoplasm. Suppose I have this cell and this cell, I'm fusing this. This is known as somatic hybridization. Okay, that is for the production of hybrid plant without sexual reproduction. Without sexual reproduction, you're doing it. So here, all of it does not make any sense. Just this guy makes sense. That is, this is the totipotency is what? The capacity to generate a whole new plant from any cell of the plant body. Now, are we totipotent, my dear children? Are our cells totipotent? Can I take Jasmine ma'am cells and make a whole new Jasmine ma'am over here? No. If someone who is not knowing about Jasmine ma'am, I am Jasmine. Okay. So, basically, certain cells of our body are totipotent. Those are the embryonic cells, okay, that I can get from the umbilical cord or from the embryonic cells. Those are totipotent. But later on that, they are not totipotent. Their potency gets reduced. Their potency gets reduced. Please remember that. So the answer is totipotency. Toti, you can remember as total. You can remember as total. They have the ability to give rise to total organism. Okay, now hint 2 always cuts DNA molecules at a particular point called recognition sequence, and it consists of, so all the restriction enzymes, my dear children, they have a specific site for cutting. They have a specific site for cutting, and over here it is asked how many base pairs are there in this particular site. How many base pairs are there? There are 6 base pairs. Please remember, there will be what? 6 base pairs, and hint 2 will be acting on these 6 pairs and cutting over here. Okay, it will be acting in a unit which consists of what? 6 base pairs. Fine, fine. Now, identify the set of correct statements. The flowers of Vallisneria are colorful and produce nectar. Now, you have to remember this. There is no trick to tell otherwise. Okay, Vallisneria is a plant that is pollinated by water. Now, water, can it see anything? No. So, does it have to be colorful? No, my dear children. So, flowers of Vallisneria colorful and produce nectar does not make any sense over here, because that is not pollinated by the insects. It is not pollinated by the insects. It is totally pollinated by water. So, this is a wrong statement. Now, the flowers of water lily are pollinated by water. You might feel that water lily is in water, so it will be pollinated by water. No. Water lily and lotus, even though they are in water, they are not pollinated by water. They are not pollinated by water. Please make it clear. So, this also would be wrong. It can be pollinated by wind and things like that, but not by water. Okay, now, in most of the water pollinated species, the pollen grains are protected from wetting. Obviously, they have to be pollen grains if they are released in water, my dear children. If they will desiccate if there is too much water in there. So, they have to be protected by some mucilaginous covering. So, in most of water pollinated species, the pollen grains are protected from wetting with the help of mucilaginous covering out there. So, this is correct statement. Now, pollen grains of some hydrophytes are long and ribbon-like. Hydrophytes are the plant that lives in the water. So, some of them, they are long and ribbon-like. Do you think pollen grains of some hydrophytes are long and ribbon-like? Yes. Now, in some hydrophytes, the pollen grains are carried passively inside the water. In some hydrophytes, what is happening? The pollen grains are carried passively inside in the submerged state only. Yes, that is also happening. Some float in the water, some goes within the water also. So, can I say C, D, E is correct? C, D, E only. I have seen an option like that. C, D, E only. Oh my god, this is A, C, D, E. Yes. So, this becomes your answer. Okay. Oh my god, wait. Oh, sorry, B is also there. Sorry, Shaktiman. Forgive me. It is correct, no? This particular statement is correct. The flowers of water lily are not pollinated by water. Yeah, exactly what I said before, but accidentally I marked differently, and I know you won't forgive me for that. Okay. So, this becomes your answer. Cool? Works now? Yes. So, don't do this mistake that I did on the day of examination. Do not be too fast. Now, given below are 2 statements. Statement 1. Parenchyma is living, but collenchyma is dead tissue. Wrong. Why? Parenchyma is living, collenchyma is also living. If you're talking about sclerenchyma, things could have been different. Okay. So, this is wrong. Gymnosperms lack xylem vessels, but presence of xylem vessels is a characteristic of angiosperm. You could have said and, but okay, anyways. Gymnosperms lack xylem vessels, yes children. They lack xylem vessels, they have tracheids and fibers and things like that. No xylem vessel. Xylem vessel is actually a it's not like they do not have xylem, okay, please understand. Xylem is a tissue made up of lots of different type of cells. In that one type of cell is xylem vessel. So xylem vessel is not there in the case of gymnosperm, and xylem vessel is there in the case of angiosperm, and that is a characteristic feature of angiosperm as well. Is this correct or not? This is correct. So can I say a statement 1 is wrong and statement 2 is correct? So with that I get 4 as the correct answer. Fourth statement is the correct answer. Yes, yes. Okay, going ahead. Formation of interfascicular cambium from fully developed parenchyma cells is an example of. Listen. Even if you do not know what is interfascicular cambium or things like that, fascicular cambium, also parenchyma, just understand the question really, really well. I will help you understand that. So, formation of interfascicular cambium from fully developed parenchyma. That means this cell is fully developed. This cell is probably what? A mature cell. It knows what it had to become. It had become that. Now, from this mature cell, you are making something else. That means this differentiated cell. I told you, whenever something attains maturity, you call them differentiated. From this differentiated cell, you are making something else. So, probably it has to undergo a process of what? De-differentiation. It has to undergo a process of de-differentiation. It has to come back to the state of like, I do not know what I want to become. And then probably it will become something else. That is interfascicular cambium. Fascicular cambium. So, this process is known as de-differentiation. Okay, it is again regaining the ability to divide. This is known as de-differentiation. Cool, works. So, formation of this from fully developed parenchyma cells is an example of differentiation? No. Redifferentiation? No. Okay, redifferentiation means coming back to the state that, okay, I have become this. De-differentiation? Yes. Okay, they are becoming something else over there. And maturation, obviously, if they had undergone maturation, there would have been differentiation, right? So, no. So, the answer is de-differentiation. Okay, it is gaining the ability to divide. Remember that. Now, inhibition of succinate dehydrogenase enzyme by malonate is a classical example of. So, there is an enzyme that is succinate dehydrogenase and it acts on the substrate. It has a substrate to act on. But what happens is that this guy comes into play. That is what? Malonate. And how does malonate actually inhibit the function? It inhibits the function by competitively fighting with the substrate for the active site. So, there is an active site on which the substrate has to come and bite. Not bite, what do you call it? Come and bind, okay, come and bind. But over here, instead of the substrate coming and binding, you have malonate which is going to come and bind. Okay, so it is competing for that. Hey, I will come, doing like that. So, this is known as competitive inhibition. This is known as competitively, it's inhibiting the substrate to come and bind. Inhibition of succinate dehydrogenase enzyme by malonate is an example of competitive inhibition. Cool, not cofactor inhibition, not feedback inhibition, not enzyme activation as well. Yes. Now, identify the type of flowers based on the position of calyx, corolla and androecium with respect to the ovary from the given figures. So, over here in A and B, my dear children, you see that you have the gynoecium over here and it's almost the same level of the androecium, that is the male part and also the flowers, floral parts which are the calyx and corolla and things like that. Okay. So, what do you call this? What do you call this condition? This is known as perigynous condition. This would be known as perigynous condition, where basically on the thalamus at the tip you will be seeing the floral parts and separately you will be finding the ovary. Both of them, they are showing perigynous. If suppose it was, I will be showing a picture like this, okay. If suppose it was like this, your gynoecium is totally covered and from this particular tip at the end of here, you are getting the corolla, calyx and things like that, is known as epigynous. Suppose opposite is happening, all of them are lower down, okay, all of them that is over here, they are lower down, but your gynoecium is the one that is superior over there, that will be hypogynous. That will be what? Hypogynous. So, below the gynous, that is gynoecium, that is what it means by hypogynous. So, over here as you can see, this and this is perigynous, which is almost like same level, and also it's at the tip of or rim of the receptor, rim of the thalamus, where all of them are easily located. So this would be perigynous, perigynous, perigynous. Okay? Yes. Now, in a plant, black seed color is dominant over white seed color. In order to find out the genotype of the black seed plant, with which of the following genotype will you cross it? I already told you, when you have something like this, listen, I cannot know the genotype of a plant. It just appears black to me. I do not know the genetic makeup. I know the physical appearance, and that is black. How do you identify whether it's like this or this? What do? You're going to cross it with what? That is the question. You're going to cross it what? That is the question. So my dear children, I am going to cross it with small b, that is the recessive parent. And what do you call this? This is known as test cross, going back and crossing with the homozygous recessive parent. Now I'll show you what is the difference that you will get when you cross this with this. Okay, you have two alleles. You have two alleles over here. This fuses with this, you get capital B small b. This with this, capital B small b. This with this, capital B small b. And this with this, again capital B small b. All of them, my dear children, would be black in color because B is dominant over this. Yeah. Now over here, let's see the scenario. Capital B small b, capital B small b, small b small b, and small b small b. Two of them are going to be what? What color? White color, because there is no dominant allele. Over here they are dominant, so two of them would be what? Say black in color. Two of them would be what? Black in color. So with this different type of offspring being produced, I understand that this particular plant that I am looking forward is not a pure plant. It is what? Heterozygous. And this particular plant is what? Homozygous. It is a pure plant. So what do I need to cross it with? It has to be crossed with small b, my dear children. Always remember, this is known as a test cross. Okay? Okay. Now, tropical regions show greater level of species richness because you have to find the reason why is it that so. So, tropical latitudes have remained relatively undisturbed for millions of years, hence more time was available for species diversification. Is it? Yes, my dear. If we compare with temperate region, they have frequent glacial glaciation happening and things like that. So it is not a constant thing that is available for them. But in tropical area, it is almost constant, and that will help in what? Species diversification. True that. Now tropical environments are more seasonal. Wrong. It's not more seasonal. It's temporary that is more seasonal. Okay, what do you have to find out? You have to find out the correct option. Yes, so this is the correct option. A is correct option. Now, more solar energy is available in the tropics? Obviously, yes. That is why it is tropical environment. Now, constant environments promote niche specialization. Yes, if you have a constant environment, then obviously that will be helping in the specialization of the niche. Okay. Now, tropical environments are constant and predictable. Yes, it is more comparatively constant and also predictable. It's not unpredictable, my dear children. Comparatively, it is predictable, right? So, can I say A, C, D, E are correct options? A, C, D, E are correct option. B is the only one that is wrong. A, C, D is the correct option, and hence your first statement becomes the answer. Okay? Yes. Now, the equation of Worldspearl logistic growth is dN by dt is equals to R and K minus N divided by K. Okay, dN by dt, you are talking about the change in the population density after time t. Yeah, where R is what? Intrinsic rate of natural increase. That is birth rate minus death rate. Simple. Okay, and N is the population size that you had, initial population size that you had. Now, K minus N divided by K, what is K standing for? So, basically, if we talk about a normal logistic growth, my dear children, if you assume that everything is there in an environment, there is no limiting factor, there is unlimited food, unlimited water, everything will be growing up like this. This is known as J-shaped curve, and this is known as exponential growth. This is known as exponential growth, right? So, over here we assume that the growth is constant, it is growing in a constant rate. But over here in natural scenario where this is being used, what is happening? You assume that growth phase is happening, but later on you understand that it cannot grow beyond a limit because everything that is there in our environment, my dear children, it is what? Limited. So, this is the capacity of growth. This is the capacity of growth, and this is known as carrying capacity. This is the maximum that the environment can carry because of the limited resources that we have. Okay, this is the limited resources. With the limited amount of resources, this is the carrying capacity. So, K stands for carrying capacity. So intrinsic rate of natural nucleus R. Biotic potential is basically birth rate and things like that. That is not a reason. Population density is denoted by N. So obviously the answer would be carrying capacity. Okay, and what is this called, this type of curve? S-shaped curve or sigmoid curve, and this is shown in logistic growth. Remember, logistic has an S, okay, and here also it's S-shaped curve, and this is exponential or J-shaped curve. Here this rate is constant, and this is shown up by dN by dt is equals to Rn. That's all. Okay, fine. Yes. Now, in the given figure, which component has thin outer walls and highly thickened inner walls? Highly thickened inner walls but thin outer walls are there. So if you are not understanding this picture, I have put this picture also. This is the labeling actually. So this is the epidermal cells, B1 is the subsidiary cells, and C thing that you see is the guard cell, and D is the stomatal pore that you have. So which one do you think has thin outer walls but thicker inner walls? As you can see in the picture, this is the one, this cell is the one that has this. Okay, what is this cell? This is the guard cell. This is the guard cell. So C becomes your answer. What is your answer? C. Your answer is C. C will be having thin outer walls and thicker inner walls out there. So yes, always remember the labelings also. Epidermal cells, subsidiary cells, and then you have these cells that are guard cells and also stomatal aperture. Fine. Next, given below are two statements. Chromosomes become gradually visible under light microscope during leptotene stage. What is leptotene stage? Lazy zebras play during the day. During the day. These are the stages of prophase 1. Prophase 1 of meiosis 1. What happens in leptotene? The chromatin start condensation, okay, and it's gradually, chromatin when it condenses, what does it form? It forms chromosomes. So can I say chromosomes are going to be gradually visible during the stage, leptotene stage? Yes, it happens. True, true, okay. Now the beginning of diplotene stage, that is this, okay, is recognized by dissolution of synaptonymal complex. Dissolution as in this, there is a complex, okay. So basically what happens, there are two homologous chromosomes like this. Homologous chromosome as in I get one chromosome from my parent, another chromosome from another parent, okay. And over here I would be having, suppose I am having gene for height, here also I would be having a gene for height. It can be a different gene, capital T, small t, but anyway I would be having it. So this is known as homologous chromosome. This would be known as homologous chromosome. So two homologous chromosomes basically are kind of joined together with the help of synaptonymal complex, okay. During this phase, zygote phase, this happens. Now slowly in diplotene phase, my dear children, what is happening? There will be dissolution. There will be removal of this particular thing over there. But are they still attached? Yes, they will be still attached at certain areas. They were there where the exchange has happened, and these areas are known as chiasmata. This area is known as chiasmata. So in diplotene stage, there is a dissolution of synaptonymal complex, but the breaking up part is really hard because still they are attached. Okay, they are still attached at certain areas, and those areas are known as chiasmata. Cool. The X-shaped structure that is formed is known as chiasmata. Fine. So this is actually correct. Both the statements are actually true. So I'll say both statement 1 and statement 2 are true. Cool. Yes, very much cool, ma'am. Fine. Children, if you are understanding, do let me know. If you are not understanding, you can ask the doubts in the comment section, which is why we have it. Yes. Now, list of endangered species was released by GAAC, WWF, FOAM and IUCN. What do you think? It was IUCN, my dear children. GAAC is related to genetic engineering. Okay, I'll talk about that. So IUCN stands for International Union for Conservation of Nature. International Union for Conservation of Nature, which is why they gave the list of endangered species. Arey, these are endangered, you have to take care of them, otherwise they will become extinct, like that. Okay. Now GAAC is Genetic Engineering Approval Committee, no, not Appraisal Committee, Approval Committee. Okay, this is wrong. Okay, approval committee regulates what genetically modified organism, as in should it be legalized or not. You have certain things, otherwise probably what will happen? We are going to pick humans and going to do some genetic modification up there. So we do not want that to happen. You get it? So basically that is why we have a committee being made like that. So there are certain rules and regulations that you have to follow. Then only you can do genetic engineering. So this is your answer. IUCN is your answer. Let me check the what do you call it? Full form first. Just like, Okay. I see the difference. Okay. So previously it was known as approval committee, but now the name has changed to appraisal committee. Okay, that is the thing. So none of us are wrong over here. I hope that question is not asked on the day of examination. If it is asked, go according to your NCERT. Okay. Now it's there in this chapter, biotechnology and its applications. Okay, last part of the chapter. Now, which of the following are required for dark reaction of photosynthesis? That is, you are talking about Calvin cycle. Calvin cycle. Now, dark reaction, the name suggests basically it can happen in dark, it does not need light. It is not like it does not happen in light, it does not need light. So, light though is not needed. And who absorbs the light? Chlorophyll. So, chlorophyll also, I'll say it's not needed. CO2, but it's needed. Because this CO2 is something that is getting fixed in Calvin cycle. Okay, CO2 is that something that is getting fixed in Calvin cycle. ATP you need, NADPH you need because energy sources are very much needed. So, I'll say 1, 2, 3 things are needed. Where do you get ATP and NADPH from? It is formed in the light reaction. It is formed in the light reaction and given to the dark reaction so that the CO2 can be easily fixed. So, I will say CDE are something that is needed for what? Dark reaction or Calvin cycle. CDE, I just have one option like that and hence your answer becomes 3. Okay? Yes. Now, the type of conservation in which the threatened species are taken out of their natural habitat and placed in special settings where they can be protected and given special care is. So, basically suppose I feel that that particular tiger is threatened. It's very risky to keep it there. So, I am taking it from that particular area and I am going to put it somewhere else. For example, a zoo or things like that. So, I am putting it in a special setting, my dear children. What do you call this? In situ conservation? Wrong. Why? Wrong. Because in situ conservation, that means you are preserving in their own area. Are we preserving in their own area? We are putting in the zoo. So, it's a wrong thing. Biodiversity conservation. Yes. Why? Actually, the answer should be ex-situ conservation. What do you mean by ex-situ conservation? Conserving organism outside their natural habitat. And under biodiversity conservation comes ex-situ conservation. Which is why this becomes the answer. Semi-conservative method, no. Sustainable development, what are you talking about? Your answer is biodiversity conservation, but if you have an option like this, please go for this. Okay, ex-situ conservation. This is for confusing you guys. Okay, so do not get confused, remember it. Now, what is the fate of a piece of DNA carrying only gene of interest which is transferred into an alien organism? I have an alien organism and I am going to put the gene of interest. So, what is the fate of that? A piece of DNA would be able to multiply itself independently in the progeny cells of the organism. Do you think this piece of DNA that I am putting will multiply itself? No, it cannot do so. It should be having an ORI site, origin of replication site, which obviously a gene of interest, if I am putting a piece of DNA, it would not be having that. It has to integrate. It has to integrate into what? The DNA. Integrate into the DNA of the host organism, then probably things could be working because this host organism DNA would be having an origin of replication and then this can replicate. So, it may get integrated into the genome of the recipient. Yes, it may. It may multiply and be inherited along with the host DNA. Yes, along with the host DNA, it may be if it is getting integrated over here. So, B and C are possible. The alien piece of DNA is not an integral part of chromosome. The alien piece of DNA is not going to be an integral part of chromosome. Please remember that. No, that's a big no. Okay. Now, it shows ability to replicate. Does it? Only if it is associated, then it shows ability to replicate. As it's written over here, it may multiply and be inherited along with the host DNA because it has integrated, my dear children, at the first place. If it is not integrated, none of this is going to happen. So, I will say B and C is the correct one. What is the fate? B and C is the fate, not any of these, not A, not D, not E. So, B and C only, this becomes the correct answer. Okay. Next thing, the lactose present in the growth medium of bacteria is transported to the cell by the action of. So, you know what, E. coli which is there, okay, E. coli which is there, it can survive on lactose also. It can survive on lactose because lactose is made up of glucose and galactose, okay. So, basically this guy can take in lactose and survive on it, but for taking this in, don't you think the membrane has to be permeable? And for making it permeable, as the name suggests, what do we have? We have an enzyme that is known as permease. We have an enzyme that is permease. Permease is going to increase the permeability of the membrane and let the lactose inside. So, it is not beta galactosidase. Beta galactosidase, what does it do? It breaks down lactose into glucose and galactose. G square. It breaks down into glucose and galactose. Acetylase, whole different function. Polymerase, baba, Does not make sense over here. DNA polymerase, RNA polymerase helps in the polymerization and things like that. Wrong. Now, 38th. Which one of the following can be explained on the basis of Mendel's law of dominance? Out of one pair of factors, one is dominant and the other is recessive. Yes, that can happen. One is dominant, other is recessive. True. Alleles do not show any expression and both the characters appears as such in F2 generation. Alleles do not show expression. They definitely show expression, my dear children. This allele showed tallness, this allele showed dwarfness, right? So they have expression. This is a wrong statement. Factors occurs in pair in normal diploid plants. Yes, they occur in pair. Factors as in you're talking about alleles only. The discrete unit controlling a particular character is known as a factor. Okay, he didn't know about genes and things like that, which is why factors basically mean genes, yeah, and he didn't know that genes are something, so he called them factors. Okay, so the genes are the one that controls all the characters in our body, right? So instead of gene, just replace it with factor. All the things are being controlled by a factor. Does it make sense? Yes. According to Mendel, yes, it makes sense. So this and this is also correct. Okay, factors occurs in pair. Yeah, and discrete unit controlling a particular character is also known as factor. Now, the expression of only one of the parental characters is found in a monohybrid cross. In a monohybrid cross, you can find the expression of only one parent, right? Because one would be dominant. So, I'll say, can I say A, C, D, E is correct? I can say A, C, D, E is the only one that is correct. I was pretty sure that B is not the correct one. So, in that way, I could exclude this, this and this. And in that way also, I could have got the answer straight. Okay, if I am very sure that this is the wrong option, then also I could have got the answer straight. That is 2. Cool. Done with 30 questions. Oh my god. Now, which one of the following is not a criterion for classification of fungi? For fungi classification, my dear children, you are going to look forward for the morphology of mycelium. You know, mycelium is made up of hyphae. And hyphae can be aseptate, septate, can have septa, cannot have septa and things like that. So, that is one factor. Mode of nutrition is not a factor. Okay, all the fungi basically they are heterotrophic, that means they depend on some other organism. They can be eating dead and decay, they can be feeding on dead and decaying thing, or they can be parasitic and things like that. So, mode of nutrition is not a let's say a parameter to classify the fungi. Mode of spore formation, yes. The fruiting body, yes. So, there are 3 things: morphology of the mycelium, mode of spore formation, fruiting body. These all are important criteria or parameters for classification of fungus, but mode of nutrition is absolutely not a criteria. Please remember, mode of nutrition does not matter how does the fungi have the food or what does it have. It is not a thing for let's say criteria for classification. Remember. Okay? Yes. Now, given below are 2 statements. Bt toxins are insect group specific and coded by gene Cry1AC. Bt toxins are insect group specific, are they? Yes, true. And they are coded by gene Cry1AC. So, there are many genes, there is only not one gene. There is Cry2AB, Cry1AB, things like that. But let us go ahead. Bt toxin exists as inactive protoxin in bee thuringiensis. However, after ingestion by the insect, the inactive protoxin gets converted into active form due to acidic pH of the insect gut. So, first statement. It has said that it is insect group specific and it is coded by a gene that is Cry1AC. My dear children, there are multiple genes, but still it is actually coded by at least this particular gene, right? At least one protoxin, one kind of protoxin will be produced by them. So, I will say this is correct. This particular statement is correct. But when you talk about this, is this statement correct? Read it carefully. Bt toxin, okay, exists as inactive protoxin. For example, if I am bacteria, in my body the toxin is being produced, but it is in inactive state. It becomes active according to this. It becomes active when an insect has it, okay. And why does it become active in the insect's body? Because there is an acidic pH in the insect gut. Is that true? Wrong. It is not acidic, it is alkaline pH. Insect gut has alkaline pH, and then this alkaline pH, the protoxin becomes what? Active toxin. This protoxin will be becoming active toxin. So, here this particular part of the answer makes it wrong. Okay, so I will say statement 1 is correct, statement 2 is false. Statement 1 is correct and statement 2 is false. 3 becomes your answer for the question. Cool. Going ahead with the next one. Bulliform cells are responsible for, bulliform cells are found in grasses, monocots, my dear children, and what do? Basically, bulliform cells, if they gain water, it makes the leaf surface open up. If they lose water, they will be coiling up the leaf. Why do we need it? Because we do not want transpiration and things like that to happen. We do not want it to lose water and things like that, which is why the coiling and things like that happen. So bulliform cells are found majorly in grasses, that is monocots, my dear children, and they are responsible for inward curling of leaves. If they lose water, they curl inward. Now, protecting the plant from salt stress, increased photosynthesis in monocots, wrong. Providing large spaces for storage of sugars, no. This is the thing. This is the thing. Now, next question. A pink flowered snapdragon plant was crossed with a red flowered snapdragon plant. What type of phenotype are expected in the progeny? Pink color. How do you obtain pink color? So, R is for red, small r is for white. How do you obtain pink color? Snapdragon or Mirabilis jalapa is the perfect example of incomplete dominance. It becomes pink when it is like this. So, you might expect it to be coming out to be red, but it's not like that. It is not completely dominant, which is why it is pink. So, this is pink. Now, it is said that I am basically crossing the pink with red. So, what is the possible phenotypes that you can get? What is the possible types of progeny that you can get? However you cross, my dear children, capital R, capital R, capital R, capital R, capital R, small r, and capital R, small r. Two of them are going to be pink and two of them are going to be red. I can find only red and pink, no white. For white, it should be small r. There should be small r from this parent also. So, red flowered as well as pink color flowers will be obtained. Only red flowers? No. Only pink color flower? Wrong. Only red, pink as well as white flowers? Wrong. There is no white. There is only red and pink. I hope you got that. Fine. Now, solve this question. How many molecules of ATP and NADPH are required for every molecule, every single molecule of CO2 fixed in Calvin cycle? Any idea? So, for fixation, the first process, okay, for the first process where your phosphate, 3-phosphoglycerate is being formed, you do not need any ATP and things like that. But from 3-phosphoglycerate, when you are making glyceraldehyde 3-phosphate, at this particular point of time, you need 2 ATP molecules and 2 NADPH. 2 ATP and 2 NADPH, okay, made it. Now, from glyceraldehyde 3-phosphate, basically, you are going to make back ribulose bisphosphate. You are going to make back ribulose bisphosphate. For regeneration, again you require 1 more ATP. So, 2 you required first, 1 more ATP you required. That means total of 3 ATP. How many NADPH? Only 2. During that process of conversion of 3-phosphoglycerate to glyceraldehyde 3-phosphate, you needed 2 NADPH, and that will be still there. That will be still there. So, your answer should be 2, 2, 2, 2, 3, 3. Okay, this is your answer. 3 molecules of ATP and 2 molecules of NADPH. 2 molecules, where do you need it? 2 molecules when you are converting from 3-phosphoglycerate to glyceraldehyde 3-phosphate. Okay, 1 where you are converting to RUBP, that is ribulose bisphosphate. 2 NADPH, same. Over here, wherever you needed 2 ATP, there you needed 2 NADPH as well. So, your answer becomes this. And with this, we have completed 35 questions. I want you to stop the video. Go just watch it like this, okay? Just go through. Pack up your brain, fix everything, and then go ahead. Until then, I will also be back by a short break. Yes, be there. Match list 1 with list 2. I hope you have done the revising stuff and right. So, list 1 is this. List 2 is slightly shifted towards this side. That is list 2, okay. So, Robert May, Alexander von Humboldt, Paul Ehrlich, and David Tillman. Who are all these celebrity people out there? My dear children, how do you find it? The easiest one to remember is Paul Ehrlich, because he was the one who gave rivet popper hypothesis. That means he considered ecosystem as an aeroplane. Ecosystem is what? Aeroplane. Now, an aeroplane is fixed together with the help of small rivets. So, these rivets are going to be the species. What? Species. So, if I remove one rivet, basically that is going to make the ecosystem go like this. Okay. Now, if I remove the rivets from the wings, what will happen? Basically, the ecosystem will anyway collapse. So, what are the rivets on the rings? They are the keystone species, the key species which are very much important. Okay. So, that was rivet popper hypothesis, and that was given by Paul Ehrlich. Okay, that was given by Paul Ehrlich. Now, how can you remember this? Basically, you can remember Paul Ehrlich has a rich in it. Okay, so he was a very rich guy. That's why he owned a aeroplane. In that way, probably you can remember. So C goes with 4. C goes with 4. I have one option like that. That's a random guess. I'm telling you. Okay. So C goes with 4, and this is how you got the answer. Let's see. A goes with 3. Robert May was the person who said that global species diversity is about 7 million. Remember this number also. 7 million is also equally important. Robert May, according to him, what is the species diversity? Global species diversity, that is 7 million. Remember that. Okay, it's a question that came in KSET examination. Now, Alexander von Humboldt was a person who gave the species area relationship. He said that basically if we explore more area, you will find more species, but it reaches to a limit. Okay, after a limit, it does not go on increasing. If I go on exploring more and more area, it won't be way too much. After a limit, it will be certainly stopping out. So, Alexander von Humboldt was a person who gave the species area relationship, my dear children. Now, David Tillman is the only man that is left. Okay, so he gave long-term ecosystem experiment using outdoor plots. So, men basically like going out. That's how you can remember. So they did an outdoor plot experiment, my dear children, according to which he said that if there is more biodiversity, it will lead to more population stability and productivity and things like that. It will lead to more population stability, as in, if for example one organism is dying, you have another organism to feed on, you have an option. So that is why it is more stable. Get it? And also the productivity also greatly increases when you have more biodiversity. That was the thing. So with that, A goes with 3. Okay, B goes with 1, and Paul Ehrlich goes with 4, and D Paul Ehrlich as in it is like that, and David Tillman basically long-term ecosystem experiment, and that's how you got the answer. Now going ahead with the next question. Identify the correct description about the given figure. So this is a given figure. What is it? It is corn cob, my dear children. It is what? Corn cob. And these things that you're seeing over here, they are tassels. What are these tassels actually? These tassels are stamen. These tassels are what? Stamen. Stamen means the male reproductive part, right? You have the what do you call, anther as well as the filament. That is what it is. Okay, they are the well exposed stamens. Why? Because my dear children, it does pollination through wind. So wind carries the pollen grains on the stamen, which is why they have to be well exposed. Like, see me, you have to carry this pollen grains. That is how it works. So identify the correct description about the given figure. Wind pollinated plant inflorescence showing flowers with well exposed stamens. This is the answer. Okay, water pollinated, wrong. Clistogamous flowers showing autogamy. Clistogamous means closed flowers. Autogamy means self pollination, as in, for example, if a closed flower has the female and the male part, it will be pollinating like this. Okay, there is no option for others to come and pollinate. That is wrong. Compact inflorescence showing does not make any sense. Over here you have wind pollinated plant inflorescence showing what? Flowers with well exposed stamens. Okay, cool. Now match list 1 with list 2. Frederick Griffith, Francois Jacob and J.Q. Monod, and Hargobind Khurana, Michelson and Stahl. Okay, so what did these guys do? That is the thing. So how do we remember this? Okay, anyways, let's start with the basic thing. Michelson, or basically we start with Jacob and Monod. Jacob and Monod, they were pretty much famous. Why? Because both of them had OO, and operon also has O. So lac operon was given by Francois Jacob and Jacques Monod. Okay, so your answer for lac operon is 4. So B goes with 4. How many options are there where B goes with 4? Only one option. I'm telling you, this is how we try to find out the answer. This is not a fun thing. Can I remember that, okay, this if I find this, I'll get the whole answer if I just come prepared like that? No. It's a very random thing that I'm doing over here. With the limited amount of information, my dear children, you will be able to get your answers if you're doing it right. Okay, this should be correct. That is the disclaimer thing that is there. So yes, Francois Jacob and Jacques Monod were the people who were behind lac operon. Okay. Now Michelson and Stahl experiment was an experiment done on Escherichia coli, and it proved the semi-conservative replication of DNA. That is, basically, if you have DNA, it will open into 2 DNAs. You have the parental strand, and there will be a new strand that will be forming complementary to it. Okay, that will be forming complementary to it. So this is known as semi-conservative. At least you have one parental strand. You're conserving half of the parental strand at least, and then you are making a new one out there. So Michelson and Stahl gave the semi-conservative mode of DNA replication. Now Frederick Griffith, what did he do? He did the transformation experiment. What experiment? Transformation experiment, where S killed, heat killed, R strain and this strain and that thing was getting transformed. Okay, and he later was trying to understand what is this thing that is making the R strain transform into the S strain. That was an experiment that was being done. Okay, but was he able to tell the genetic material mediation? He was not able to tell. It was Avery, MacLeod and McCarthy who came again and said that DNA is the transforming principle. But that also was not recognized until and unless who came? Hershey and Martha Chase came. Okay, Hershey and Chase came to do their experiment, and that's how it was proved that DNA is the genetic material. So Frederick Griffith was the person who gave the transformation principle experiment. Okay, and Hargobind Khorana, we are left with what genetic code? So Hargobind Khorana helped really much in the formation of what? Genetic code. And with that we get 2 as the answer. A goes with 3, A goes with 3, B goes with 4, yeah, and C goes with 1, and Michelson and Stahl goes with semi-conservative mode. Cool? Yes. If you didn't understand the Frederick Griffith transformation principle part, you can tell me. I'm not explaining the whole thing because it will take a huge lot of time if I give on the explanation for that. Okay. So yes, spraying sugarcane crop with which of the following plant growth regulators increases the length of the stem, thus increasing the yield? Auxin, gibberellin, cytokinin and abscisic acid. So growth promoter hormones are 3: auxin, gibberellin and cytokinin. This is the growth inhibitor hormone. Okay. Now among them, which is the one that helps in the lengthening of the stem? Increasing the length of the stem. It is gibberellin. It is what? Gibberellin. Gibberellin has L in it and it is there for lengthening. You can remember like that. Gibberellin for lengthening. Okay, not cytokinin. Gibberellin for lengthening. Gibberellin is the one that is used for lengthening of the stem. Please remember it. Come on, you have to come this side. Yes. Next. Match list 1 with list 2. Last 10 questions. Okay. Rose, pea, cotton and mango, twisted aestivation, perigynous flower, drupe and marginal placentation. So if you talk about marginal placentation, that is the easiest spot that I remember. So if you talk about a pea plant, you have placentation here at one particular margin. Okay, this is where the placenta is left. So I'll go with P. P is marginal placentation. So B goes with 4. How many options do you have like that? 1 option. Again, I got the whole answer because I knew 1 option. You get it? But can you take this risk always? No. You have to be, as I said, you have to be pretty sure about the one option that you're looking forward for. If you're not, you mess up the whole thing. So that is why having an understanding of the concepts is really much important, my dear children. Do not take it lightly. Okay. Now, okay, we have mango. So mango goes with drupe. It is a proper fruit, fleshy fruit that we have. So it was drupe. So D goes with 3. Now cotton, if we talk about cotton, my dear children, it is having twisted aestivation. It is having twisted aestivation. What do you mean by that? I have a picture over here. So see the cotton flower, okay, the petals, they are like one over the other, it is located like that. One end will be below, one end will be above, one end will be below, one end will be above. Similarly, as you can see over here, this is a cotton flower that you have. This is known as twisted aestivation, arrangement of the petals out there. Okay, so cotton with twisted aestivation. What about rose? Rose is perigynous flower. Perigynous, as I already told you, what is perigynous? Basically, you have what? Perigynous, I had the picture of perigynous, right? We'll come to that. Where is the picture? The picture is gone. Yes, this is perigynous. Where basically on the thalamus, at the tip of the thalamus or receptacle, you have all the petals and things like that, and the ovary, androecium and gynoecium is almost up to the same level, my dear. This is known as perigynous. So basically, rose has perigynous ovary. Remember that. Rose has perigynous ovary. And I wanted to tell you something. We were talking about hybridization somewhere, right? We spoke about somatic hybridization. I was explaining you something. So somatic hybridization is not the protoplasm that fuses. Basically, we call it protoplast. Okay, we call it specifically protoplast. Okay, wait, let me come back to the question. Yes. So protoplast is basically when I am removing cell wall from the plant cell, what I am left with is known as protoplast. Now, protoplast consists of protoplasm and things like that is there. Okay, but its exact term to be used is protoplast in somatic hybridization, not protoplasm. Okay, protoplasm is actually cytoplasm plus nucleoplasm, I know. But apart from that, you have cell membrane also. So including cell membrane as well as the cytoplasm as well as the nucleoplasm together, you call this as protoplast, just removing the cell wall. So just removing the cell wall and making them fuse is somatic hybridization done with the help of protoplast. So answer for this question is what? 1, my dear children. I hope you got it. So rose is showing perigynous flower, okay, P is marginal estivation, and cotton is twisted estivation, and marginal placentation, sorry, and cotton is twisted estivation, and mango is drupe. Okay, yes, I hope these pictures are also helping you well. Now read the following statement and choose the set of correct statement in the members of Phaeophyceae. Phaeophyceae, and you're talking about brown algae. Okay, now asexual reproduction usually occurs by biflagellate zoospores, is it? Yes, there will be 2 flagella, and that is a zoospore. Asexual reproduction occurs with the help of that. Now B, sexual reproduction is by oogamous method only. No, it can be oogamous, it can be isogamous and isogamous and things like that. Okay, oogamous basically where both of them are different in shape, isogamous same in shape and size and things like that. Okay, motile, non-motile, you had different methods. It's not just oogamous. Now this is wrong. Okay, now you have to find out what is correct, right? Yeah, stored food is in the form of carbohydrates, which is either mannitol or laminarin. Correct, very much correct. The major pigment found are chlorophyll A and C, carotenoids, and xanthophyll. Yes, xanthophyll, carotenoids, the orangish pigment, xanthophyll is the yellow brown pigment. So obviously in a brown algae, obviously it will be there, yeah. And chlorophyll A and C is also there. Chlorophyll A and C, carotenoids, and xanthophyll. Now vegetative cells have a cellulosic wall, and usually covered on the outside by gelatinous covering of algin. Yes, it is there, which is why it is like little bit jelly like towards the outer layer. Okay, so I'll say A, C, D, E, all of them are correct. A, C, D, E only, this would be the correct answer. B is the wrong statement because it is not the only oogamous method. There can be multiple other methods as well. So this becomes your answer. Okay, remember. By this you remember the features of Phaeophyceae: asexual reproduction by biflagellate zoospores, my dear children, and stored food is basically mannitol or laminarin. Major pigments is A and C and carotenoids and xanthophylls. Okay, vegetative cell, they have a cellulosic cell wall. Apart from that, my dear children, they have gelatinous covering of algin. Okay. Now identify the step in tricarboxylic acid cycle, that means TCA, or what do you call it as citric acid cycle, okay, which does not follow oxidation of substrate. Which of this step does not follow oxidation of substrate? What do you mean by oxidation of substrate? Basically you're going to borrow something from NADH or things like that, and that is how things work. So malic acid to oxaloacetate, here obviously oxidation is happening. Okay, succinic acid to malic acid, oxidation is happening. Succinyl CoA to succinic acid, wrong. Here oxidation is not happening. Isocitrate to alpha ketoglutaric acid, oxidation is happening. I'll explain you. I have a thing for explanation as well. Where is it? Did I actually remove it? So my dear children, all of the places, my dear children, they are taking the electrons. Okay, they are taking oxidation. Basically means what? Taking up of this thing. Reduction basically means giving hydrogen. Reduction basically means you're getting hydrogen, not giving hydrogen, you're getting hydrogen. That is why you say you're reduced. Oxidation means basically you are giving the hydrogen ions, or basically you're talking about electrons in that sense. Either way, but over here in malic acid to oxaloacetate, yes, oxidation of substrate is happening. There will be substrate that is oxidized. Succinic acid to malic acid, yes, again it is happening. Isocitrate to alpha glutaric acid, it is happening. Succinyl CoA to succinic acid, over here GTP is being formed. Over here GTP is being formed, and here oxidation is not happening. Substrate level phosphorylation is happening. Okay, substrate level phosphorylation is happening, my dear children. Okay, the substrate is helping in the, what do you call it, in the formation of GTP, that is substrate level phosphorylation. So oxidation is not happening. I don't know where did I lose that. Yes. Now given below are two statements. In C3 plants, some oxygen binds to rubisco, hence CO2 fixation is decreased. Is it? So rubisco enzyme, my dear children, has an affinity for oxygen as well as CO2. If oxygen is there, it will go bind to the oxygen, or else it is CO2. So in C3 plants, there is high chance, okay, there is high chance that oxygen can come and bind, which is why CO2 fixation could be reduced. That is absolutely true. Now in C4 plants, mesophyll cells show very little okay, when this happens, when rubisco binds to oxygen, my dear children, Okay, what happens, what do you call this, is photorespiration that will be taking place. So in C3 plants, photorespiration can take place. Now in C4 plants, okay, mesophyll cells show very little photorespiration. That means photorespiration is happening in them according to the statement. Okay, so in C4 plants you have two things, you have mesophyll cell and you have bundle sheath cell. You have mesophyll cell and you have bundle sheath cell. Okay, now over here I am saying mesophyll cells show very little photorespiration while bundle sheath cells do not show photorespiration. Do you think here photorespiration would be happening? No. Mesophyll cells are designed in a way that the CO2 is actually fixed over here. CO2 is fixed over here, not with the help of rubisco. Not with the help of rubisco, but by PEP carboxylase. So if rubisco is not there, I don't think photorespiration would be happening over there. Rubisco is right over here. Rubisco is within internal layer. Okay, it is inside, and this CO2 which is fixed come back over here, again gets fixed over here. Okay, this CO2 is fixed in the form of oxaloacetate over here, and this oxaloacetate converts into malate. Malate comes inside the bundle sheath cell, breaks that back into CO2, and then the CO2 gets fixed again. This is how things work. So since rubisco enzyme is not present in the mesophyll cell, there is no chance of photorespiration to happen in the mesophyll. So here little photorespiration is absolutely wrong. While bundle sheath cells do not show photorespiration, that is absolutely correct as well. So this statement is wrong, this is correct. So in total, if you say, it is going to be wrong only. So can I say this statement is wrong and that statement is correct? Statement 1 is correct or statement 1 is true, but statement 2 is what? False. Cool? Yes. Now next, match list 1 with list 2. List 1, GLUT4, insulin, trypsin, collagen cells. Now even if I do not know anything, I can understand from the options that I have, hormones, enzyme, intracellular ground substance enables glucose transport into the cells. So GLUT4 probably has something to do with glucose. So it enables glucose transport into the cells, my dear children. Yes. Okay, so A goes with 4. Can I find my answer with that? I just have one option with A with 4 and this becomes my answer. B, insulin is what? A hormone which is secreted by the pancreas. It is produced by the pancreas. Trypsin is what? Trypsin is an enzyme that breaks down what? Peptides, my dear children, that break down proteins into peptides. Now collagen cells, collagen, what is it? It basically it is act a ground substance. Between the cells you have collagen, making them bind together. Okay, so collagen is between the cell, you call it intercellular ground substance. So 1 becomes your answer for this particular question. Match list 1 with list 2. So GLUT4 with enables glucose transport into cells, insulin, hormone, trypsin, enzymes, and collagen, intercellular ground substance, which is right over there. Yes. Okay. Let's go ahead. Match list 1 with list 2. List 1 and list 2. Monodelphis, diadelphis, polyadelphis, epiphyllus. So what do you mean by monodelphis? Monodelphis means there is only one thing on which your stamens and everything is being located. There is only one unit of one bunch of stamen which is there. That is monoadelphis. Where do you find it? In the case of China rose. In the case of China rose, you might have seen there's so many things like this. So all of them are on one bunch, my dear, and they're forming just one bunch. This is monodelphis. So I'm pretty sure about China rose. So A goes with 4. With that, can I find the answer? No. I have 2 options like that. So and I can eliminate at least 2 options. Okay. Now diadelphis. Diadelphis means what? Diadelphis means you have 2 bunches. You have what? You have 2 bunches. Sorry, it does not look like this. There will be a bunch like this and probably a bunch like this. Okay, so there are 2 bunches of what? The stamens out there. So this is diadelphis and it is found in what? Can you guess? Can you guess? Okay, I'll keep it there. Polyadelphis. Polyadelphis, that means they have multiple bunches, and that is found in citrus, So C for polyadelphis and C goes with 1. C goes with 1. I have one option like that, and this becomes my answer. So A goes with 4, C goes with 1, and B. What about B? Diadelphis is actually seen in pea plant. In what? Pea itself. And epiphyllus is basically found in lily. Okay, epiphyllus basically means what? Epiphyllus, any guesses? If someone knows about epiphyllus, what does it mean? Can you put it down in the comment section? Let me see who all knows about epiphyllus thing. It is seen in lily. Okay, so this is your answer. Monodelphous means one bunch, didelphous means two, polydelphous means multiple, epiphyllous means you have to tell me. So monodelphous is found in China rose, didelphous in pea, and polydelphous in citrus plants. Citrus always have polydelphous. Now, okay, the DNA present in chloroplast is, so I hope you know that DNA is present in chloroplast as well as mitochondria. Okay, we have it. Yes. So what is the type of DNA that we have? Usually in normal cells, in our normal nucleus, okay, what is the type of DNA that we have? It is linear. It is what? Linear. But in our mitochondria or in plants chloroplast, my dear children, you have circular DNA. You have circular DNA and they are double stranded. Always remember. So DNA present in chloroplast is linear double stranded? No. Circular double stranded? Yes. So it is believed that chloroplast and mitochondria, they used to be certain organisms which basically came inside the eukaryotic cell and became a part of it, became an organelle inside that. So they were separate organisms who had their own circular DNA and things like that, later came into eukaryotic cell and then forever it stayed there. So it has circular and double stranded DNA. It's not just chloroplast, also mitochondria. Now, in an ecosystem, if the net primary productivity NPP of the first trophic level is 100x kcal meter per year, okay, what would be GPP, gross primary productivity of the third trophic level of the same ecosystem? So you are given the NPP of the first trophic level. What is it? NPP. NPP is what? Net primary productivity. NPP is what? GPP minus respiratory losses, right? So it is a total amount that you can give to an organism. It is a total amount. After you did all the things like that, there is a total amount that you can give to the next organism. So I will say GPP of this is of the first trophic level. This is of the first trophic level, that was 100x, right? So GPP of this would also be 100x. I hope you're getting it. Listen to me carefully. GPP minus R, that means its production minus its usage, will be NPP, and that is given to be 100x. It is given to be 100x. Now this total amount is given to the next, because that is after loss you're talking about it, right? So this total amount is given to the next organism. So for the next organism, this will be the GPP. For the next organism, this will be the GPP, and it will be minusing it, and then you get NPP. Okay, but how do you minus it? Basically, you can say that 10% of it is lost. 10% of it is lost. How do you calculate? So basically, if you calculate, you will get 10x. So 10x will be NPP for second trophic level. Okay, after loss, it is the only thing that is left to give to the next organism. So now, the next organism for the next trophic level, that is third trophic level, what will be the GPP? What is present over here? It is 10x. It is what? 10x. And what is the question over here? The question is the same. In an ecosystem, is the net primary productivity of the first trophic level is 100x. And net primary productivity of the first trophic level is 100x. That means after minusing, you are getting 100x. Then what will be the GPP of the third trophic level? So for GPP of the second trophic level, you are going to calculate 100 only, because that is the available thing. You're going to minus it. Okay, how do you minus it? You're going to say calculate 10% of it. Only 10% of it will be gone. Rest of it will be lost, right? So you'll say calculate 10%, you get 10x. Now this 10x, which is NPP, is the whole thing that is available for the next organism. So that is the GPP of that. So 10x would be your answer. I know this is kind of confusing, but this is the answer. So your answer becomes 10x, and the unit you have to keep it as such. Okay, now if you're someone who is finding difficulty, I don't know where I put the answers. Okay, I got it. I didn't send that file. Anyways, chalo. Which of the following are fused in somatic hybridization involving two varieties of plants? First thing, children, if you're not able to understand it still, you can tell me. I'll try to explain it once more in the comment section. Okay, but try to understand it. But try your level best to understand it by your own. It's gross primary productivity minus respiratory losses, that is known as NPP. NPP as a whole is available for the next organism. So for the next organism to offer to the next organism, it will be acting as a GPP. It is what it received. Now it will be utilizing something for itself and giving off the next thing. What it gives us is what? 10x. Now 10x would be after the other organism get it. From this 10x, again you will minus and give it to the next organism. That will be NPP. You get it? So NPP of one will be GPP of another. Okay. Which of the following are fused in somatic hybridization involving two varieties of plants? I told you, in somatic hybridization it is protoplast. It is protoplast. Suppose this is a cell, this is also a cell with the cell wall. Sorry, the cell wall is this, the cell membrane is this. Okay. So basically what I am going to do is I am going to remove the cell wall and make it fuse. So this is a protoplast and this is a protoplast. Protoplast has cell membrane, also cytoplasm and also nucleoplasm. Also cytoplasm and also nucleoplasm. So this becomes your answer. Okay. Not callus. What is callus? Basically, if I am cutting a part of the plant, okay, I am growing it, giving what nutrient media and things like that. This part which I am going to grow, okay, this is known as calyx. Calyx would be basically I am taking a plant part, okay. This is known as explant. It is an explant. It is not a part of a plant, it is now explant. I am putting in a nutrient medium, it will be growing. It will be growing, right? This is now known as callus. Now callus is put in growth hormone medium. It will be having shoot, it will be having root and things like that, and then it goes on. That is callus. Pollen, so you know, somatic embryos, no, the answer is not that. It is just the protoplast. Now, match list 1 with list 2. Citric acid cycle, glycolysis, electron transport system, proton gradient. So glycolysis, my dear children, we all know it occurs in what? Cytoplasm. What is glycolysis? Conversion of glucose to pyruvate. So glycolysis occurs in the cytoplasm. Can we find the answer with that? B with 1, there is only one option, and yes, we found out the answer with that. Now A, citric acid cycle, TCA cycle. So the pyruvate which is there, it gets into the mitochondria, converts into acetyl-CoA, right? And the acetyl-CoA gets into the citric acid cycle. So where citric acid cycle happening? It will be happening in the mitochondrial matrix, inside the mitochondria's matrix region, okay? Now electron transport system. Electron transport system is located in the inner mitochondrial membrane. Mitochondria has two membranes, inner mitochondrial membrane, outer mitochondrial membrane. In the inner mitochondrial membrane, you would be having the electron transport system. Okay. Now listen, this is the matrix. This is where TCA cycle is happening. Okay, tricarboxylic acid cycle and things like that. Oh my God, TCA. Now here ETC, electron transport chain. Okay. Now this is the outer membrane, right, that you have. So in between this, what you have the proton gradient. The protons are being pumped from here to here. So here in between you have the proton gradient. So this is known as intermembrane space. Okay, intermembrane space. Cool. And yeah, that's all. So citric acid cycle or TCA is happening in the matrix of the mitochondria. Electron transport is the inner mitochondrial membrane, and proton gradient is in the intermembranous space of mitochondria. So this becomes your answer. Okay. Yes. Now, last question, the easiest of all of them. Which of the following statement is correct regarding the process of replication in E. coli? DNA-dependent DNA polymerase catalyzes polymerization in one direction, that is 3' to 5'. The DNA-dependent DNA polymerase catalyzes the polymerization in one direction, that is 3' to 5'. No. Always remember, DNA polymerase, RNA polymerase, or even translation happens in one direction, and that is 5' to 3'. 5' to 3'. So DNA dependent DNA polymerase catalyzes polymerization in 5' to 3' direction. This becomes your answer. DNA dependent DNA polymerase catalyzes polymerization in 5' to 3' as well as 3' to 5'. Wrong. Okay, and over here DNA dependent RNA polymerase is categorization. Wrong. In DNA replication, why do you need RNA? Okay, so and the direction itself is wrong. So this becomes your answer. And my dear children, I hope I have I should not be showing that. Yes. So I thank you from the bottom of my heart if you have been with us throughout this whole video. I think I could help you. If there are certain things which I could not explain you can put it down in the comment section. I am here to rectify those things. I am here to explain you those things. Do not feel judged. I won't be judging you for anything. Please just ask me the questions that you have. Yeah, I will be very happy to help you out. And my dear children, if you're liking this video, do not forget to like and also share with your friends and also subscribe to the channel. You all are going to ace your NEET examination, and if you have still not enrolled into the free mock test which is there, please enroll it right away. The link will be given in the description box. Do check it out as well. Wishing you all the best. You guys are going to do amazing. Bye-bye. Take care. Lots of love.